Plant tissue culture. Cutting and grafting are the improved methods of natural vegetation propagation in plants. In 1902, G. Haberland noticed that the plant cells can be grown in synthetic media. The discovery by Haberland that the plant cells have the capacity to grow in a nutrient medium in presence of sufficient light made an impact in plant propagation and crop improvement. The ability of a plant cell to give rise to a whole plant is called totipotency. The cell and tissue culture techniques have taken the method of asexual reproduction in plants. In this method, instead of taking cuttings from a plant, a cell or a few cells or a tissue is taken from a plant and cultured in suitable containers in which nutrient medium is present. The portion of the plant that is taken as the desired plant is referred as explant. By this method, thousands of plants can be produced from a single desirable parent in one generation. These techniques require maintenance of sterile conditions in the medium. The explant needs to be sterilized before it is introduced into the medium. The medium is kept free from bacteria and fungi. Cells or tissues are grown in culture under a set-like condition. In the tissue culture, the explant often divides to give rise to an unorganized massive tissue called callus. The callus or the explant is such will undergo differentiation into roots, shoots, or embryo-like structures is called embroids. This differentiation is dependent on the concentration and combination of the plant's growth substances like auxins, cyanins, gibberellins, etc. In the medium, plants raised through tissue cultures are routinely used in agriculture, horticulture, and forestry. We'll use a diploid explant. We get a diploid plant like in any other vegetative propagation methods. Similarly, if we want to develop a haploid plant, it is possible to obtain it through tissue culture. Development of haploid plants through tissue culture was discovered by Indian scientists Shipra Guha and Satish Maheshwari.